Welcome. Today, my colleagues and I will be presenting on the use of bioacoustics techniques to study vocal behavior of threatened Southeast Asian vertebrates. Southeast Asia is home to numerous endemic and at-risk species. Using passive acoustic monitoring, or PAM, we studied 4 of these species from the island of Borneo. PAM requires minimal human intervention and is ideal for inaccessible regions like the rainforest. We used autonomous recording units, or ARUs, to capture these animals' vocalizations. Vocalizations are central to understanding vertebrate communication and can be studied from the individual to the population levels. This presentation uses spectrograms, which are a visual representation of sound, with frequency shown on the y-axis and time shown on the x-axis.
First, we'll be discussing individual variation in the calls of female Bornean gibbons. Gibbons are pair-bonded arboreal apes that rely on vocalization to defend their territory and communicate with each other. Previous work has shown that we can acoustically distinguish between over 30 individual gibbon females with 95% accuracy. We are interested in how the slight differences observed in the great calls of individual females can be used to identify them via passive acoustic monitoring. Our experiment was performed using playbacks of different individuals' calls, which were then recorded at various distances. Among the questions we asked were, how are individual signatures transmitted? Does classification ability change throughout the day? And do different parts of the call contain individual signatures that travel better in a forest environment? More specifically, is there a difference between the introduction and the trill of the great call? First, we found that as the distance from the playback increased, our ability to classify individuals decreased substantially. We also noticed that the time of day did not influence the classification accuracy of individuals, though that data is not pictured here. Finally, we determined that the trill portion of the gibbon's great call generally provided a higher classification accuracy than the introduction, despite the fact that the trill degrades faster over distance. This may provide some insight into their respective functions, although further research is likely necessary to determine exactly what those functions are.
Next, we turn to the population level study of the great argus. The argus is a near-threatened pheasant that lives in thinly forested areas at elevations below 1,200 meters. We set up 10 recorders for 2 months across roughly 2¼ square kilometers of the Danam Valley Conservation Area. This contained between 7 and 32 territories. We investigated the questions, is there spatial variation in long calls? And do population-level patterns of variations exist? Further, we were interested to know if unsupervised methods can be used to study potential individual-level differences. First, we found that there was spatial variation in argus calling events. Next, we created a biplot of principal components analysis. Each point here represents one argus call. The color represents unsupervised clustering classification. There was evidence for clustering, which we interpret as potentially being differences among individuals. Future research is needed to validate this unsupervised method.
PAM can also be used in interspecies comparisons. In our last case study, we look at the differences in vocal behavior of helmeted and rhinoceros hornbills. Both hornbill species are similar with regards to their diet, size, and breeding behavior. Their calls also show substantial overlap in frequency. Both calls are composed of a series of short notes that become more broadband at the end of the call. However, since they are easily distinguishable on the spectrogram, we can use PAM to compare their calling behavior. Our data is displayed in this figure, which shows the frequency of helmeted and rhinoceros hornbill calls throughout the day. Both species are strictly diurnal in their calling behavior, and we found no evidence of temporal niche separation. We also investigated the spatial partitioning of hornbills by recording their calls at 5 locations. We found no evidence of spatial partitioning between the species. However, the hornbills may be able to maintain niche separation due to differences in feeding location.
In summary, for our individual level study, we investigated variation in the great calls of female gibbons and how individual identification varied with environmental conditions such as time of day. This has raised further questions about the calling behavior of gibbons.
For our population level study, Unsupervised clustering provided evidence for 32 unique clusters or individuals, but we need to validate this using ground truth data.
And finally, for our interspecific study, we found no evidence of spatial or temporal separation in helmeted and rhinoceros hornbill vocalizations. In the future, we can use more fine-scale acoustic monitoring to find out where in the territory the hornbill species are calling from.
In conclusion, PAN can provide important baseline information related to the behavioral ecology of at-risk populations.
We'd now like to thank the following organizations for their support. Special thanks to the Cornell Lab of Ornithology for funding our research this summer. And finally, thank you to everyone in our audience for watching.